11 a.m. EDT. So I'll let Matt take it away. Welcome to Potpourri 2.
Thank you. So hello, everyone. I'm talking on the evolution of bats and whether it, or more to the point, nightbirds, whether their evolution was curtailed or impeded by bats. And by nightbirds, I'm not referring to owls, but the nightjar group, often known as Caprimulgiformes. So here's a phylogeny. We've got the Caprimulgiformes here in yellow, and Caprimulgiformes include, well, a number of different families, and here's a few pictures for you all. They're a paraphyletic order, or formerly order, and so you can see that they start off for early evolution as nocturnal insectivores, and there's a shift to diurnal, primarily insectivores, at least among the swifts, but also the hummingbirds, and this is a group together known as the Apodiformes. This phylogeny is reasonably well set now. A few little changes here and there, but it's pretty much become a consensus, at least among molecular phylogeny. The earliest fossils from this group tend to jump around a little in the phylogeny, but perhaps more importantly, their age is roughly around 55 million years old, and it's several different fossils at roughly that time, so the beginning of the Eocene, or close to the beginning of the Eocene. Really special preservation is required, for fossils of this sort. And so it's quite possible that they may be slightly older, or that there may be a few members that are missed in the fossil record that are a little older. I'd say the Strysores group, their fossil record's pretty good, particularly during the Eocene, from about 55 million through to maybe 34 million, and then it tends to fade away. One question is whether that's because of fossil preservation issues or whether it's because of limited diversity at the time. So now looking into bats, this is a phylogeny, a dated phylogeny, some work I did with Carmelo Fricciano a few years ago. And basically it comes down to the different families of bats, various representatives here. So we found that the bats diverged from other mammals at about 65 million years ago, and that they have a crown origin, or the origin of the modern group, at about 54 million years ago. There's a bit of argument in the published literature about just how old that crown group is, varying from about 50 to 65 or 66 million years. But after we removed a few biases in the molecular dating and looked at the calibrations fairly carefully, we're reasonably happy with this age. And it also fits quite well with what's known from the fossil record. And I guess one thing about that also is about those arguments is that the crown origin of bats is not so important for this question of whether there's potential competition between bats and nightbirds. That crown origin might have been isolated somewhere and wouldn't necessarily have any impact on the evolutionary potential of another group. So what's more important is their global spread, potentially for impeding Strysores diversification. And we can see that by looking at the fossil record. The earliest fossils of bats are basically filling into all continents within just a few million years. So Europe, Asia, North America, South America, not Antarctica, but all the others, between 55 and 51 million years ago. There's also quite a rapid diversification at this time. So I wanted to work out or try and estimate the divergence times within Strysores birds. So use Prüm et al.'s top 10 data set, samples all the Strysores families. It's a reasonably large data set, but not so big that we couldn't manually align it. Manual alignment versus automated alignment was actually what this project was originally about. And so this is something of a side issue. We added in a few additional genes that are commonly used, RAG1, MYC, and Site B, maximum likelihood phylogeny and IQ tree, divergence estimation, and MC tree within the PAML program. Here's the phylogeny or dated phylogeny of the Strysores that we came up with. So the origin or crown origin of Strysores just after the Cretaceous-Paleogene boundary, about 55 million years ago. So when dinosaurs and so forth became extinct. So their original diversification might have been niche filling with the extinction of quite a few bird groups and obviously pterosaurs as well. So lots of diversification very early, within a million years or two. Then after that, we get the divergence of the apodoforms, so swifts and hummingbirds, from Strysaures, or the other, in this particular case, the owlet nightjars. Then a break in diversification until about 40 million years ago. Then a few of the major groupings diversifying a little later. So then a lineage through time plot for Strysaures to take a look at this relative to bats. And what we can see here is that really rapid early diversification, and then no more diversification among Caprimulgiformes until quite some time later. This little reddish or orangey coloured dot here, that's actually divergence of apodoforms, hummingbirds and swifts, from Strysaures. So that may or may not really count. So then wanted to look how the anatomically modern volant or flying bats fit relative to this picture. So what we see here is this is that range of those fossils on different continents that I noted before. And it could be a little bit older if there's fossil preservation issues. And so the molecular dated stem lineage of bats is indicated here in terms of their timeline. So we'd expect that bats evolved in terms of flying and they're increasing their distribution or dispersal somewhere between about here and here. So right in that area or that time span in which there's a lack of diversification in nightbirds. So we wanted to take a further look at this and see how it might be reflected potentially in climate. And so this is global average temperature, which we can see is pretty warmish when Strysaures are diversifying really early, rapid diversification when it's warmish. But then the world heats up quite a bit through the Paleocene-Eocene thermal maximum and beyond. And it's in this period here with a much warmed world in which there's no diversification of Caprimulgiformes or nightbirds. And that's when the bat fossils are found. It's also when there's this rapid diversification of the early bat lineages. So I guess the other thing to note here is that then Strysaures or the nightbirds are not diversifying again until we get over this humping temperature and get cooler temperatures once again, and then they diversify a bit more rapidly. So this reminded me a little bit of a study from Brian McNabb quite a few years ago, way back in 1973, where he was able to look at the energy budgets of in particular, vampire bats, and see that he can model those energy budgets, and that fitted their ranges, their geographic range, really quite well. So if I expand out a little bit further, there's been a lot of debate on diversification and extinction drivers, and it's centered on climate versus biotic interactions, largely. And this has been dubbed the court jester versus red queen hypothesis, with court jester being the chaos of climatic effects, primarily, and other environmental effects. It's a coined term by Anthony Barnosky, championed by people like Mike Benton, whereas the red queen was more about biotic interactions, and that's coined by Lee Van Valen. I think what I'm doing here really is primarily just noticing patterns with respect to birds and bats. There's a lot more work to be done on this, but from what I'm seeing, there's at least potential to consider the possibility that these diversification dynamics between bats and birds, or bats and Strysaures, nightbirds, might be an emergent property of both. So climate change shifting the balance of evolutionary competition. And by evolutionary competition, I don't see that as mutually exclusive of ecological competition, but as potentially more about long-term access to evolutionary opportunity and ecospace. So that's basically it from me. A couple of acknowledgements, a picture of some little red flying foxes that I took recently. I did take a photo last night of a frogmouth, but it's so poor I decided I like the stamp version better. Okay, thank you very much.
Thank you, Matthew. We have definitely plenty of time for questions. So feel free to raise your hand and we'll call on you, or post them in the Q&A in the chat. All right. You wait for someone to be brave enough to ask a question. I guess I'll jump in with one. Is there kind of a smoking gun evidence of potentially still ongoing exclusion between bats and nightjars? Do you see any patterns of regions with greater bat diversity have reduced nightjar diversity or something similar in just the modern day?
Yeah, look, I haven't looked into that as much as I perhaps should have. I only got that phylogeny finished off 10 hours ago. So there's more work to be done here. However, I think there's a latitudinal gradient to this, and that kind of fits the temperature effects as well. So I wouldn't go so far as to say a smoking gun, but there is some more general sort of evolutionarily um, sort of like evolutionary competition rather than ecological, I suppose, or ecospace distinctiveness between bats and birds. That seems to have held up for a long time. One way that that can also be looked at is through the extinction and diversification dynamics in terms of the probabilities that they would have the diversity that they have given null models, and those null models don't fit particularly well. So it does look like something more is going on. Hard to rule out other factors, though.
So there are a couple questions in the Q&A. The abstract mentions ameliorating molecular substitution rates based on life history correlates in the divergence time analysis. Do you mind expounding on that?
So it seems that diversification time analysis doesn't do particularly well sometimes when you've got very large and very small animals in the same phylogeny, or for that matter, looking at um, longevity is another life history trait that seems to correlate with rates of evolution. So what I did with that was I removed the really large, like ostriches and emus and really large and long-lived birds, and that resulted in I suppose, dating that lined up a bit better with fossil records. One of the mammal papers I cited earlier was me and Carmelo Fruciano did that with mammals as well, and so it's that method is actually in that paper, which is I think 2018.
Another question that's in a similar vein to the one I asked. Do you see any potential clues towards this interaction of theories in regions where these species ranges overlap? Is there significant niche partitioning, competition, or anything like that?
I think my expertise is simply too far away. I think I'm happy to leave that to the ecologists and evolutionary ecologists. Um, yeah, it can be difficult to distinguish these macroevolutionary patterns and look at their origins at the very short time scales.
All
But
right.
I'm sure you have expertise on that than me.
Was echolocation in sync with flying abilities? I'm not sure I fully understand that question. If
Oh, okay.
if Maria wants to.
Some of those I'm not sure exactly what the question is, but some of those very early bats, including stem bats, are showing morphological correlates of echolocation. So it does seem to have been something that came in quite early with bats.
All right, well, if there's no further questions, feel free to stop sharing slides, Matthew, and we can get the next speakers set up. But if you have any other questions, feel free to paste them in the Q&A. I'm sure Matthew will be happy to answer them. Feel free to share, Rock. Yeah. All right, take it away, Rocco.
Good morning and good afternoon, everyone. Today I will present the results of my current work where I've been looking at the signatures of selection acting on a key protein involved in innate immunity. In the context of host-pathogen interactions, the red queen dynamics predict that hosts and pathogens engage in cyclical adaptations and counter-adaptations in response to each other, but that there is no long-term winner of this race. And a well-known group of genes found to be involved in these dynamics are the immune genes. Very well-studied example of these immune genes are those immune genes that directly interact with the pathogens. An example are those receptors that recognize and bind to the so-called pathogen-associated molecular patterns, or PAMPs. So upon binding and recognition of these PAMPs, the immune response is then triggered. So these interacting gene pairs engage in coevolutionary dynamics, and particularly these immune genes have a remarkably higher polymorphism compared to other genes in the genome and are often found under balancing selection. Famous examples of these receptors and very well studied are the major histocompatibility complex and the toll-like receptors. Another class of immune genes are those that interact with the self, with the host cells, in what has been referred to as the missing self immunity, which means that in this case, the immune response is triggered against those cells that are lacking some molecules that instead are present in the host. But I will explain this better later on. These genes have been underrepresented in the literature because they are commonly expected to be monomorphic and to have a constrained evolution because they interact with host molecules, and therefore we can expect that any mutations might be deleterious. However, these genes are often the target of immune evasion, whereby pathogen produce effectors that can bind this immune regulator. And in this way, they can manipulate the immune response and evade clearance. So they're basically saying, I am one of the good guys. So as I said, not many studies investigated the selection acting on these genes, except one exception are the killer receptors, sorry. One example of this regulator of missing self-immunity is the complement factor H, or CFH for friends. CFH binds to molecules called polyanions that are present on the host cells of all vertebrates. Instead, bacterial cells do not present polyanions, and therefore they don't bind CFH. In the absence of CFH, we have the activation of the complement cascade, whereby several proteins sequentially and with cascade reactions bind to pathogens and eventually lead to their lysis. In the host, instead, CFH inhibits the complement activation. That's why we say that it's a regulator of missing self-immunity. However, CFH is also the target of immune evasion by many pathogens. One such pathogen is Borrelia, which is the causing agent of Lyme disease in humans. And here on the left, you have a photo of Borrelia. So Borrelia produces on its surface, produces many transmembrane proteins. One example is OSPE, which are able to bind to CFH. And by covering its surface with CFH, Borrelia can effectively inhibit complement activation, therefore evading immune clearance. So we can expect that CFH is under selection to avoid binding from OSPE. However, selection on CFH might also be constrained by its need to bind to host molecules. However, no study so far has investigated selection acting on CFH. So the aims of this study are to identify signatures of positive selection along CFH, to investigate polymorphism in domains interacting with Borrelia, and to analyze population differentiation and structure compared to the genomic background. As a model system for the host, we chose bivalves, European bivalves, because these animals are reservoir hosts for Borrelia. which means that they significantly contribute to the natural circulation of Borrelia. We collected ear samples from 13 populations across Poland for a total of 154 samples. And we chose populations where previous studies identified different bivalve clades or ecotypes in the northeast, in the west, and in the south, with also some populations in the middle. So with these samples, we conducted the genotyping by sequencing for CFH, and we also conducted RAD sequencing, with which we obtained roughly 60,000 biallelic SNPs randomly sparsed across the genome. Moving on to the results, we firstly conducted the N over the S ratio tests across the CFH gene using PAMEL and MIM models to identify signatures of positive selection. Here you have a schematic representation of the CFH gene with all its 22 exons. The brown lollipops show non-synonymous SNPs, while the yellow stars show the codons with evidence of positive selection according to both PAMEL and MIM. So as you can see, we found many non-synonymous SNPs across the gene. However, many SNPs are concentrated in 3 domains, CCP6, CCP19, and particularly in CCP20. Specifically, in CCP20, we have codons that directly interact with Borrelia, here in red. And as you can see, either directly these codons or the codons in their proximity are under positive selection. Therefore, we then focused on this domain, CCP20 domain, because it directly interacts with Borrelia and because it presents the most majority of SNPs, with roughly 1/4 of the non-synonymous SNPs present in this domain. So based on this domain, we found that CFH is very polymorphic, with 6 variants identified across our populations. And based on their phylogenetic similarity, we grouped these variants into 3 main clusters. And as you can see that these clusters are also structured among our populations, and they reach near fixation in different locations. We have cluster 2 in the northeast, cluster 3 mainly in the west, and cluster 1 mainly in the south, with evidence of some admixture in the populations in central areas. The structure mirrors that identified with all the genomic RAD-seq markers. Therefore, now we question whether the structure that we observe for CFH is stronger and whether gene flow in CFH is reduced compared to the genomic background. To do this, we conducted genomic scans of FST outliers, and we used Bayesian models via the software BayPass, which we run using the RAD-seq markers, which we call our genomic background, and the 16 SNPs that we identified in the CCP20 domain of CFH. Here we have on the x-axis, in this graph, we have on the x-axis the scaffold and the genomic positions, and on the y-axis, FST equivalent. The red line identifies a significance threshold, which represents the 99% quantile obtained with simulated data. And any point above this line represents a SNP that is significantly differentiated among populations. In yellow, we have the 16 SNPs inside the CCP20 domain of CFH. And as you can see, 13 of these 16 SNPs are significantly under diversifying selection. We also identified 2 RAD markers that were lying inside the CFH gene. Here are identified in dark blue. And one of them is under diversifying selection, kind of confirming the results that we obtained for the CFH SNPs. Therefore, these results show that the structure that we observe in CFH is stronger and gene flow is reduced compared to the genomic background. So in synthesis, we found that CFH is highly polymorphic, despite this immune gene interacts with host molecules. The codons that are hijacked by Borrelia are under positive and diversifying selection. These results support the idea that CFH engages in coevolutionary dynamics with Borrelia, and that there is local adaptation in CFH, possibly to local Borrelia populations. Our study is also the first one to the best of our knowledge, to identify non-neutral polymorphism in CFH. And now we set out to investigate whether population structure and signatures of selection in Borrelia's OSP, to confirm sorry. We now set out to investigate the population structures and signatures of selection in Borrelia's OSP to confirm this coevolutionary hypothesis. And with this, I thank you for your attention and I welcome questions.
Thank you so much for that talk, Rocco, and we definitely have time for questions. So please raise your hand or type them into the Q&A. I'll again kick it off, I guess, while people type. What's known about Borrelia prevalence across this entire range? Is there any relationship between any of the particular markers and I know you're investigating next the actual Borrelia variants, but maybe one of these variants that you found is just found in places where there is basically no Borrelia, so it's managing to protect the mice from it.
So we found Borrelia almost everywhere, although the prevalence in bivalves is not super high. We have among our collected samples, maybe 20% in each population, usually on average, are infected. But ticks do present Borrelia. So Borrelia is, for those of you who don't know, is a vector transmitted pathogen. So it's transmitted to hosts by ticks. So the ticks are indeed infected with Borrelia almost everywhere.
I will follow up, I guess, if there's no other questions. So in a lot of these post-parasite coevolutionary stories, I think it's really good that you narrowed in on a gene that immediately makes sense because there's just so many gene pairs to actually compare. I saw there's other places in the genome that you're seeing differentiation between the populations just in the RADseq. Do any of those seem to pop out also to immune genes? Are there other candidates popping out here that seem to be differentiated between the populations?
Yes, we do have. We actually have other components of this complement cascade, which are C3 and C5. We have other immune genes. I think one of them was interleukin 6. We have a few more, which we probably we will see if we will follow up with further studies. But it's definitely interesting to see that there are other components of the complement because especially CFH interacts with C3 and C3 interacts with C5. So, that can make up an interesting story and it will be interesting to investigate that.
All right. Well, if there are no further questions, I guess we can stop sharing, Rocco, and we can slowly transition over to Agnes. Again, if you have come up with a question, feel free to post in the Q&A. The speakers will be able to answer them down the line. Yeah.
Okay, are you seeing my screen?
We can see your screen.
Should I wait a minute?
Yeah, we should wait to try and start right at the turn. In case someone from a different section tries to hop in last second.
Yeah, just tell me when to start.
All right, feel free to start.
Okay, perfect. Well then, good afternoon, morning, evening, wherever you are, and very happy to talk about a recent study I did together with my, let's see, I need to click in the presentation, here we go, with my PhD student, Konstantin Kopper, about how shifts to novel environments triggered pollinator shifts in a large tropical plant family. And I know that not everyone is familiar with plants here and pollination, so I'll start pretty broadly. And among botanists, as myself, we usually consider shifts among functional pollinator groups as an important source of flower diversity and reproductive isolation. So basically as an important mechanism in plant speciation. This is exemplified here. You see 3 different flowers in each panel. Each flower comes from a different family, and the most closely related flowers here, for the case of the plant family Caryophyllaceae, look quite different. So these red flowers are most closely related to these purple flowers adapted to bumblebees and to these white flowers adapted to hawk moth pollinators. And so the idea is that different pollinators can exert strong selective pressures on flowers, on flower phenotypes, and shifts among such pollinator groups are an important source for adaptive divergence. What we know much less about, however, is why pollinator shifts happen in the first place and when they happen in the evolutionary history of plant lineages and also where they happen. When we switch from an evolutionary perspective to an ecological perspective, then we may find some answers to these questions. I've pulled out a study from the European Alps here, which is one of many studies that have documented major turnovers in pollinator communities across environmental or also latitudinal gradients. In this case, you see a lowland plant pollinator network represented here, where different orders of insects are represented by different colors. And as we move up along the gradient to highest elevations, we see pollinator networks that are mostly dominated by flies, so Diptera. What we don't learn from these ecological studies is whether there are consistent patterns across lineages. And at an evolution conference, I guess everyone's interested as to whether anything that we see in nature is replicated in evolutionary time. In an attempt to look at these environment-pollinator relationships in a more comparative way, a couple of years ago, we put together a data set of more than 2,000 plant species across 26 different plant clades, evolutionary independent lineages, and found a very strong association with elevation specifically, in that in the tropics, insect pollinated species tend to occur at lower elevations than their vertebrate pollinated relatives. Now, this raises the question whether such pollinator shifts are potentially a prerequisite for the colonization of tropical mountains or a result thereof. And to date, there's no studies that have looked in depth at the evolutionary sequence of pollinator shifts or climatic niche shifts, which we've just before heard a little bit about the Corchester or red queen scenario, and both may apply to lineage diversification of plants. Now, when we want to look at the evolutionary sequence of environment and pollinator shifts, I've drawn out a few hypothetical scenarios here where we compare ancestral and novel pollinators and ancestral and novel environments. And we may envision different scenarios. For example, environment shifts could precede pollinator shifts. Pollinator shifts are to our left, environment shifts to the right, and we see environment shifts at deeper nodes in the phylogenetic tree than pollinator shifts. Alternatively, we may observe pollinator shifts preceding environment shifts, or correlated evolution of the two, in that shifts in pollinators and environments occur at similar nodes or similar ages across a phylogenetic tree. The system that I'm using now to look into these questions is the tropical plant family Melastomataceae, which ranges among the 10 largest plant families worldwide with more than 5,800 species. It counts with at least 11 independent shifts into mountain environments and at least 21 independent evolutionary pollinator shifts. So it has this evolutionary replication within the large family. To look into the pollinators, we start with the odd pollination systems. Approximately 5% of species have shifted pollinators. Pollinator shifts are mostly recent in the group and along the tips of the tree and have given rise to small clades. In 6 tribes, we see shifts to different vertebrate pollinators, including hummingbirds, bats, and rodents. In 1 tribe, we see 3 independent origins of passerine bird pollination, where passerine birds feed on sugary food bodies provided by the flowers. And in 1 tribe, we see at least 3 independent shifts to generalist insect pollinator assemblages, where more than 80 different insect taxa have been observed foraging for nectar and pollen in these flowers. Okay, that's 5% of the 5,800 species. The majority of species, so the ancestral pollination system in the group, is pollination by bees. Actually, a pretty cool pollination system that is called buzz pollination, where bees vibrate flowers in order to extract pollen from the flowers. So this is the widespread, our ancestral condition that we're dealing with. Now, to investigate the evolutionary sequence of environment and pollinator shifts, we've pulled together a data set of more than 300 Melastomataceae species, covering all 22 currently recognized tribes and all genera. We had empirical pollinator observations for 250 of these species, which still is not all. We used machine learning methods, random forest analyses, so basically classification techniques, which we trained on species with empirical pollinator observations and flower traits, and then used those refined models to predict pollinators for the species for which we don't have empirical pollinator observations, which led us to 53 species that are classified as shifted in this data set. We then went ahead, collated GBIF occurrence data, climatic niche data, and started reconstructing the evolutionary history of pollinator shifts and elevation, or elevation evolution. Just to get back to our baseline expectation, as in the study that I mentioned before, also in this plant family, we see that bee pollination occurs at significantly lower elevations than vertebrate pollination. So we have this basic pattern that there seems to be an ecogeographic stratification in pollination strategies. Now, when we look at the ancestral character estimations, we see the pollination to the left and the elevation to the right. And I know that it is small. That's why I've pulled out or zoomed into a few clades. We do see that elevation elevation shifts to higher elevations above 1,000 meters have occurred at deeper nodes than pollinator shifts represented in orange here. Same holds when we look at a different clade, where again we have strong support for elevation shifts preceding evolutionary pollinator shifts. This holds with different bindings for elevation. Obviously, elevation is a continuous variable. It's difficult to classify into mountain or not, or montane or not. So we ran it with elevation at 500 meters as a cutoff for montane, or cutoff at 1,500 meters for montane. And this pattern does hold across different delimitations of what is montane and what is not. So we do find very strong support that elevation shifts did indeed precede pollinator shifts and likely were a trigger thereof. When we think a little bit, this again supports it basically looking at the different nodes in the phylogeny and the reconstructed probability of having shifted from bee to vertebrate pollination or non-bee pollination and having colonized mountains. If across the phylogeny the trend should be towards colonizing mountains before shifting pollinators, we would expect nodes that are descending from shifted tips to aggregate here with a higher or earlier shift probability for elevation than for pollination. And this is exactly what we find with all nodes descending from shifted tips aggregating along the y-axis of this plot, not along the x-axis. And now I'm jumping to the ecology of pollination. It makes absolute sense that we see this happening in tropical mountains. May be obvious to all of us. As we move up in elevation, temperature decreases naturally. It becomes rainier, windier, colder. And these are conditions that are not favorable in general to ectothermic pollinators, such as most insects, and endothermic pollinators, which can maintain their own body temperature, such as hummingbirds, bats, can much more easily deal with these adverse montane weather conditions. There are notably some bee groups, such as bumblebees, which can actually maintain their own body temperature and which are well-adapted high-elevation pollinators. They are not as diverse in the tropics as they are in temperate zones. But this might be an explanation for why we see another interesting pattern when we zoom into the phylogeny again, and that is that not all environment shifts lead to pollinator shifts. So in this group here, we have multiple instances of mountain colonization, but they go without pollinator shifts, which raises the question, why do environment shifts not always lead to pollinator shifts? And there are multiple hypotheses that might explain this pattern. One might be that montane bee-pollinated species occupy more bee-friendly habitats or areas within the mountain environment than their vertebrate-pollinated relatives. To look into this, we again compared abiotic climatic niche variables for bee-pollinated and shifted species. This time we're looking at mean annual temperature. This is for the complete data set, basically mirroring the plot you saw before for elevation, since elevation and temperature are strongly correlated. And this now is just zoomed in for species above 1,000 meters, comparing bee-pollinated species and shifted relatives. And we see that within this montane environment, delimited here as anything above 1,000 meters, bee-pollinated species seem to occur in slightly warmer areas, warmer mountains, maybe southern slopes of mountains, than vertebrate-pollinated species. We can do the same if we just subset the data set shown here in panels C and D to bee-pollinated species, contrasting tribes which have shifted pollinators and which have not, but just looking at the bee-pollinated species. And there we actually also find significant differences below 1,000 meters. Bee-pollinated relatives of shifted species do sit even in warmer niches than their purely bee-pollinated or bee-pollinated relatives from just bee-pollinated tribes. And within the montane environment, again, bee-pollinated species of shifted tribes tend to sit in slightly colder niches than their bee-pollinated or than bee-pollinated species from purely bee-pollinated tribes, which means that there's some indication that these montane species that have sometimes shifted pollinators, sometimes not, have generally evolved into slightly colder high elevation niches, but the bee-pollinated species did stay in slightly warmer niches. Another explanation that we may think of is, I need to start the video, let me see. When we look into the actual pollination, and I couldn't help sharing this beautiful video of a bus pollinating bee at work. This is how they vibrate the flowers. They grab the stamens and then start applying the high frequency vibrations in order to extract pollen from the flowers. And one aspect that might explain why some bee-pollinated species can retain their ancestral niche in high elevation ecosystems is that they might evolve traits that maximize pollen export or pollen transfer in these environments. And such a trait might be an increase in pore size. The stamen pores are the area or the only part of the stamen where pollen can come out, which the bees actively collect. And we see stamen pore sizes ranging from below 200 micrometers to more than 500 micrometers, so quite a big range. And an increase in pore size might increase pollen release rate when bees apply vibrations. So when we compare pollen area along elevation, we do indeed find a positive increase of pore area or pore size with elevation, indicating that high elevation species do indeed have larger pores, likely facilitating the pollen release, even with scarce bee pollinators at high elevations. So to conclude, are pollinator shifts a prerequisite or a result of mountain colonization? Our data strongly suggests that mountain colonization precedes pollinator shifts, and hence
Great, I
pollinator
have
shifts
to
are
cut
a
you
result.
off, Agnes. The next speaker has to start in 15 seconds. So we'll have
15
to
seconds.
cut you off.
Okay. Well, I
All
think
right.
I've said it all.
All right.
Thank you.
If you could stop sharing, and Mariangela, if you can swap over. If you have questions for Agnes, please type them into the Q&A. Go ahead, Mariangela.
So, can you see the presentation?
Yep.
Okay. So, good morning, everyone. My name is Mariangela Iannello, and I'm a postdoc at the University of Bologna. And today I'm going to talk about mitochondrial segregation in animals. So, as you know, a proper segregation of the genetic material during cell division is essential because it prevents the insurgence of genetic incompatibilities or the development of disease. Concerning nuclear DNA, we know that the mechanisms of chromosome segregations are more characterized. But concerning mitochondrial segregation and mitochondrial DNA segregation, these mechanisms are much less understood. Now we know that mitochondria are organelles in cells. They're present in multiple copies and they possess their own DNA. And these organelles are not static, but they undergo events of fission and fusion, and they are essential for many biological processes. So, for example, ATP production, calcium regulation. Sorry, I'm trying because I cannot see. Okay, all the slides. Okay, thanks. Calcium homeostasis, apoptosis regulation. So a not proper segregation of mitochondria can have severe consequences because we can have the spread of mutant phenotypes. And for example, during embryo development, the segregation in some blastomeres of dysfunctional mitochondria can lead to developmental arrest. So the mechanisms are not fully understood. A lot of studies so far focused on yeast because here we have asymmetric cell divisions. And thanks to this model, we now know that at least in this species, we have a complex remodeling of the cytoskeleton, particularly actin filaments, that allows an active transport of mitochondria from the mother to the daughter cell. But concerning, for example, mammals, it is believed that a stochastic mitochondrial segregation happened during embryo development, and it seems that mitochondrial fragmentation may have a role in ensuring fair distribution of mitochondria in different cells. So it seems a passive mechanism, but it is also true that in mammals as well, we can see events of asymmetric segregation of mitochondria. So, for example, it seems that stem cell inherit younger mitochondria compared to somatic cell. So it means that there is an active segregation and selection of mitochondria. So the mechanisms are not clear in mammals, but in animals in general, and this possibly reflects the lack of a suitable model to study mitochondrial segregation. And so in this context, bivalves can help perhaps to have new evidence about this process. So we have that in around 100 of bivalve species, we have a different mechanism of mitochondrial segregation, and this is called Doubly Uniparental Inheritance of Mitochondria, or DUI. And in this species, what we have is that paternal mitochondria segregate in the male germline blastomeres. So we have paternal mitochondria only in males, while females have the maternal mitochondria. So just to let you know that this mechanism is poorly understood in general, so we don't know why if there is some advantage in this mechanism, how many times it evolved, when it evolved, or in general the molecular mechanisms of this process. But concerning mitochondrial segregation, here you can see that we can actually see during embryo development that paternal mitochondria that are stained here in with mitotracker green show a different segregation in male or female embryos. So in male embryos, you can see that they stay aggregate and eventually they will segregate in the germline blastomeres. But in females, they show a dispersed distribution. So this is a unique condition in animals where we can have two different patterns of mitochondrial segregation. So we can take advantage of this kind of mechanism, possibly to identify players involved in mitochondrial segregation. So another thing you should know about DUI is that this differential pattern of mitochondrial segregation is believed to depend on factors deposited on eggs. And that's because first, this differential pattern of segregation happens very early in the development, so before the zygotic genome activation. And also because it has been seen that this pattern of segregation, as well as the sex of the embryos, depends only on the mothers. So we have females that can produce up to 100% of eggs that will develop females with this pattern of mitochondrial segregation. And there are males which will produce, females that will produce eggs which will develop up to 100% percentage of males with the aggregate pattern of segregation. As you see, there is a link between the sex of the embryos and the pattern of mitochondrial segregation. And but at least for the species, Mytilus galloprovincialis, which is a DOI species, it seems that these two different phenotypes are linked but not causally linked. So as I mentioned, the aim of this work was to take advantage of this model and investigate genes involved in mitochondrial segregation in animals. So what we did was to identify females that produce mostly females and females that produce mostly males, and we collected from different samples female biased eggs and male biased eggs, and we investigated differential transcription among these conditions. So I will focus on differential transcribed genes for time reasons. So we found around 200 differential transcribed protein coding genes, of which most of them show an upregulation in female biased eggs, and only 4 show an upregulation in male biased eggs. So the aim was to try to understand if these differential transcribed genes have a role in the different mitochondrial segregation that we see. But as we mentioned, we have also a link between the pattern of mitochondrial segregation and the sex of the embryo. So we investigating genes involved in mitochondrial segregation or genes involved in sex determination, which, by the way, is unknown in bivalves. So for this work, we were interested in genes involved in this DUI-specific pattern of mitochondrial segregation. To try to select genes involved in this process, we tested if these genes may have a different evolution in DUI species compared to species with strictly maternal inheritance of mitochondria, or if they interact with genes that show a different evolution in DUI species. And for this reason, we performed molecular evolutionary analysis. So for this analysis, this is the dataset that we used. These are genomes and transcriptomes from 36 species of bivalves, and 10 of these bivalves show doubly uniparental inheritance. You see them in blue. And from this species, we use transcriptome and genomes to identify protein coding genes and to identify orthologs. So at the end of our analysis, we ended up with almost 9,000 single copy orthogroups. And so what we wanted to see if there were genes in DUI species showing similarities in terms of rate of protein evolution beyond those expected by chance. And so identifying the genes, it means that possibly they could be involved in the DUI trait. And for doing this, we use this program, Structure, who basically analyze the branch length on each pairwise comparison between DUI and not DUI species. And these are our results. So we identified, here you can see the distribution of p-values of our results. And in blue, you can see the distribution of p-values for genes showing convergent constrained evolution in DUI species. And you can see for constrained genes that we can see an enrichment for low p-values compared to what we expect using instead random controls here in gray and in pink. But for accelerated genes, actually, we don't see this pattern. Instead, the level of genes with low p-values is lower than what we could expect by chance. So we removed these genes from our analysis. And we also tested if these genes with a convergent constrained evolution show also a different DN over DS in DUI species compared to species with strictly maternal inheritance of mitochondria. And for this analysis, we used a CodeML. And we found that for 206 genes, we have a stronger purifying selection in DUI species compared to SMI species. So this seems to be genes that have a different convergent evolution in DUI species. So going back to our question, do differentially expressed genes evolve differently in DUI species compared to SMI species? Well, the answer seems no, because the overlap between these two sets of genes is very low, and this is something that we could expect by chance, this number of overlap. So the answer is no. And do these genes, differential expressed genes, interact with genes showing different evolution in DUI species? And in this case, the answer seems yes, because we identified the protein-protein interactions across these genes, and the number of interactions is higher than what we could expect by chance. And it is also higher considering the differential expressed genes alone and differentially evolving genes alone. And if we take a look on some statistics, we can see that genes showing convergent evolution in DUI species, which here are shown in blue, they usually show a higher level, higher degree and stress, which are metrics that usually measures that the genes may be key components of the network. And if we take a look at the annotation of these genes, we can find different motor proteins or different proteins associated to macrotubule-based movements, which are genes and pathways known to have a role in mitochondrial segregation. And here you can see some enrichments of the biological processes identified. So we can find organelle organization, cytoskeletal organization. So these are clues that possibly this network is involved in the pattern of mitochondrial segregation. And if this is true, possibly we can take advantage of this network to identify some candidates involved in such a process. So for example, in violet here, you can see two genes which are upregulated in male biased eggs, which are samples that we will have aggregate mitochondria. And these two genes are actually both activators of DRP1, which is a master regulator of mitochondrial fission, which seems in mammals to have a main role in allow a proper segregation of mitochondria during embryo development. And another interesting gene is this MAT2A, which seems very central in the network, but so far this is just a speculation because we know that this gene have a role in preventing the onset of cell division until chromosomes are properly aligned. And I don't know, our hypothesis may be that this gene may have a role in preventing also the onset of cell division until perhaps a proper segregation of mitochondria happens, but we don't know, but the take-home message here is that hopefully this model system, BUI, may provide a new perspective for studying mitochondrial segregation in animals. And we believe that the integration of different analysis, so for example, differential expression, differential transcription, protein-protein interaction, hopefully allowed us to have a more complete view of the process. And hopefully this network highlight the complex interplay across these proteins and highlight new candidates. I think I'm running out of time. So just to conclude, and thank you all for the attention.
Thank you for that talk, Mariangela. There are no further talks in this session, so we have a little bit of time for Q&A, but if you need to get to a different talk, feel free to leave something in the chat. Mariangela can try to answer later.
Okay.
I'll start with a question of, it's fascinating system, especially I hadn't heard of the elements. I've heard of doubly uniparental inheritance, but I didn't realize that you had these biases depending on the maternal phenotype in terms of male, female offspring. You said you don't really have any idea of what the sex determining mechanism is, but as you're teasing this apart, are there any interesting hints of like what are the actual differences between these male or female producing?
Yeah, we have no clues, but actually this is our next project because we are trying to investigate, part of the result of this result could answer the question, what can underlie sex determination, because these female bias eggs versus male bias eggs. So we are going to take a look at this, and also we are going to perform single cell experiments on embryos, so male embryos versus female embryos. So hopefully we can try to have some clues about this mechanism in the next experiments.
All right, well, if there are no further questions, thank you all for attending this session. Hope you have a great rest of the conference. I hope to see you in some of the other sessions there.